Hi, everyone. Welcome to today's webinar. My name is Heather Fears, and I will be your host for today's webinar event. So I would like to start off by going over today's agenda. So first, we're going to have the welcome, and I'm going to introduce the speakers. Then we're going to turn it over to the speakers for their main presentation. We're going to have a Q&A session after that. Could you please submit any questions through our chat feature while our speakers are talking so that we do not interrupt them? Then we will go into our promotion events, and then we have a surprise announcement. So thank you again for your participation. Today we are honored to invite Dr. Paige Vinson and Dr. Sixue Zhang from the Southern Research Institute to deliver a wonderful lecture on AI-powered HTS, unlocking precision on hit discovery. And Dr. Vinson received her Ph.D. in analytical chemistry from Emory University. Early in her career, she worked with Thermo Fisher Scientific as an application scientist and product manager in their laboratory automation division. She then began working at Vanderbilt University as an in vitro pharmacologist and eventually became the director of their high-throughput screening facility, as well as an assistant professor in the Vanderbilt Center for Neuroscience and Drug Discovery. Currently, she is with Southern Research as the director of high-throughput chemistry. She is a part of a broader drug discovery community through her involvement with the Academic Drug Discovery Consortium, where she serves as president, and with the Society of Laboratory Automation and Screening, where she is the co-chair of the Screen Design and Assay Technology Interest Group. With over 20 years of experience in both academic and industry settings, Dr. Vinson has a deep understanding of the drug discovery process and continues to make significant contributions to advancing scientific discovery. Dr. Zhang earned his Ph.D. in computational chemistry from the University of Illinois, Urbana-Champaign. He is currently with the Southern Research Institute, where his team plays a critical role in utilizing AI models developed with in-house data to quickly identify potent compounds that would otherwise take years to discover. In addition to his leadership role at Southern Research, Dr. Zhang also serves as an advisory member for the Center of Clinical and Translational Science at the University of Alabama, Birmingham. With over 10 years of experience in computer-aided drug discovery, Dr. Zhang has collaborated on more than 50 drug discovery projects across various therapeutic areas. His work focuses on leveraging AI and the next generation molecular modeling to streamline and accelerate the discovery of novel therapeutics. And now I will turn it over to Dr. Vinson and Dr. Zhang.
Thank you, Heather. So I guess this is the slide that we'll start on.
Oh, sorry. I'll introduce let me introduce what we will learn in this webinar. So we will learn how AI-driven technologies enhance throughput screening for faster drug discovery, how combining silico and high-throughput screening methods can enhance hit identification, hit to lead and lead optimization stages of a drug discovery program, and the requirements of applying this approach along with examples of successful implementations. Now I will turn it over to Dr. Zhang and Dr. Vinson.
Okay. Thank you. Let me quickly share the slides. Okay, we are ready to go.
Okay, is everybody seeing the slideshow? Great. Well, thank you for that introduction, Heather, and hello, everyone. Dr. Sixue, Zhang, and I are looking forward to sharing our experiences in combining our HTS and in silico approaches to inform early drug discovery, and we'd like to thank MedChem Express for the opportunity to do so.
And today, Dr. Paige Vinson will give us an overview of the HTS approaches for drug discovery, especially the hit discovery. And then I will discuss about the in silico approaches, including artificial intelligence for hit discovery. Then we will see how we can combine those two approaches together to make even better results. And lastly, we would also love to hear your feedbacks.
So first, we'd like to share a bit about Southern Research, where Sixue and I work. We are a nonprofit research institute founded in 1941, located in Birmingham, Alabama. It's the highest NIH-funded non-academic research center in Alabama and has a $150 million annual economic impact. And over the course of our Institute's 80-plus year history, Southern Research has developed over 20 clinically tested agents, including 7 FDA-approved cancer drugs. During COVID-19, we contributed significantly to vaccine and therapeutic development, conducting more than $30 million in research. And our facilities include 150,000 square feet of wet lab space, including ABSL-3 facilities and large animal housing capacity. And we're excited that we are building a new flagship research building, which will add 117,000 square feet of lab and office space to our campus, and that's slated to be completed in the summer of 2025. So I also wanted to start with a short review of the history and capabilities of the Southern Research HTS Center, because this has helped to lay a foundation for our AI-accelerated HTS approach. So we were established in 2001 and served as the screening center for infectious disease for the Molecular Libraries Screening Center Network and the Molecular Libraries Probe Production Centers Network. And during this time, we interrogated 47 targets in 171 assays, in addition to supporting internal programs. And over the last decade, our center has performed over 100 campaigns with over 380 distinct assays and over 5,500 experiments, testing over 22.5 million samples. And from 2020 through 2023, we contributed to multiple COVID-19 therapeutic programs with cell-based screening in a BSL-3 environment. And as a result, we have over this 20-year history, or over 20-year history, we have curated a rich internal bioassay database. And the Southern Research HTS Center consists of several functional groups, including compound management, assay development, and screening and informatics. We've made significant investments in technologies and material assets, such as compound libraries. Our collections total over 750,000 compounds and consist mostly of diverse drug-like collections, but we also have some smaller bioactive collections as well. And although our initial focus during the MLSCN and MLPCN times was infectious disease, we have grown increasingly disease agnostic in order to support the growing needs internally and externally from our internal scientists and our external collaborators and clients. And therefore, we have experience across different target types and multiple assay modalities. But the HTS Center is only one part of our integrated drug discovery platform at Southern Research. So as shown on this slide, we have several groups, including computer-aided drug design that Sixue is the head of, medicinal chemistry, structural biology, and support for ADME-Tox and DMPK studies. And these all work together to support the internal expertise in therapeutic areas of oncology, infectious disease, and pulmonary disease, as well as the needs of external collaborators and clients. And a recent development is the establishment of programs to support early commercialization efforts and precision medicine. And through various funding mechanisms, including federal, state, and foundations, along with fruitful collaborative efforts, we have grown a robust drug discovery pipeline with programs ranging from hit identification through clinical testing across multiple therapeutic areas. And it's worth noting that a number of collaborations we have that we've had with the University of Alabama at Birmingham have been funded through a state-funded accelerator program, which was mentioned on the previous slide in terms of early commercialization support. So most of these programs that are listed here have benefited from contributions from both HTS and in silico efforts. So I'm sure that the graphic or some variation of this graphic on this slide is familiar to most of you watching this webinar. It's a high-level summary of the drug discovery process and some of the activities that occur in each stage, starting with target identification, going through clinical trials. And within this process, the application of high-throughput approaches has the purpose of aiding drug discovery programs in the identification and characterization of compounds with the desired pharmacological activity. And we use automated approaches to allow high capacity and high throughput testing to do that. And although we usually think of this occurring during the hit identification stage, as shown in the third chevron on this slide, we can actually benefit from using high throughput methods across stages whenever possible. And this helps to provide rapid feedback to chemistry efforts, and it increases the capacity for testing. This is an important consideration when you are incorporating in silico approaches, because this can result in an increased demand for bioactive data. And we even strive to automate more challenging translational assays, especially as things like organoids and different 3D models are becoming more popular. We're trying to automate even more complex assays that are important to programs and that can inform them during even sort of later stages, going from lead optimization into preclinical development. So to reflect this sort of growing impact of high throughput approaches at Southern Research, many times we call it what we do high throughput discovery. So we're not limiting what we do to only hit the identification in large screening campaigns. So whether you're screening for hits or informing programs at later stages, there are several requirements for operating in high throughput. Of course, you need a source of test articles. These can be small molecules, peptides, antibodies, RNA drugs, etc. Although what we are focused on today will be small molecules. This source of compounds is actually very much at the heart of the combination of HTS and in silico methods, and I'll touch on that again in the next few slides. You need a robust plate-based assay that represents the biology of interest, and depending on the demands and stage of the program, the throughput of the assay may vary, as I mentioned previously about automating more complex assays. And although fully robotic systems are not required, automated liquid handling and plate-based detection are required. So continuing on through this loop, another crucial aspect is an effective informatics group that can provide the data analytics to present the high volumes of data in a form to allow data interpretation and decision-making. This is an aspect that I think sometimes we overlook, and it's really important. But of course, lastly, we also need experienced scientific and support personnel to design and execute the experiments. So I wanted to go through some considerations for HTS as applied in the hit identification stage, if we're, focusing on that. And just on its own, without supplemental methods, HTS has value and merit in this case, because one, it enables an unbiased and rigorous approach. We're not making any assumptions about chemical structure, especially if we are screening a diverse chemical set. It provides experimentally confirmed hits for chemistry follow-up, and it requires a well-performing assay that represents the desired biology that you're looking to affect. Of course, it only finds a starting point for optimization, and that's where we are with any screening technique, whether it's traditional HTS or in silico. We still are just finding starting points for developing a drug. One of the things I would consider a limitation is that we're limited to million scale chemical space. So there are only so many compounds that you can physically screen for a campaign due to time and cost. And so that brings us to this slide. This graphic helps to illustrate the relative chemical space that different methods have the capacity or capability of testing. A commonly quoted number for all chemical structures is 10 to the 60th. So when testing with conventional HTS, depending on the collections that are being screened, this has an upper limit in the low millions. DNA-encoded libraries, which is an approach that has gained popularity, can provide access to a larger billion-scale chemical space. However, its application, at least for now, is limited to mainly finding binders to targets as opposed to measuring functional consequences of target engagement. Therefore, a high-throughput approach is maybe necessary for follow-up after a DNA-encoded library screen. And so in silico methods have a clear advantage in this case, in that it has access to trillion scale chemical space through large databases containing these structures. So it's able to access these virtual synthesizable databases to give us access to that space that we otherwise would not have access to. So at this point, I'll turn it over to Sixue to provide more details of the models he has developed and applied to our programs.
Thank you, Paige, for the nice overview of the HTS approaches. So as Paige mentioned before, we have an integrated drug discovery platform. Besides HTS, there are also other functional teams in the platform, including the in silico teams. So that's why we always try to find a way to collaborate with each other, see if the in silico method can further help with the HTS effort. And in terms of hit discovery, there are two types of in silico approaches we often use for our projects. They are the target-based virtual screening and the phenotypic-based virtual screening. So target-based virtual screening refers to the prediction of the test article with the specific interactions with protein structures. Usually it's the therapeutic target protein. And of course, the advantage of using the target-based virtual screening is that by doing that, we can more likely ensure clear molecular mechanisms of any hit come out of this effort. However, if you only use a target-based virtual screening only approach, then there's also the weakness. For example, some hits that are active against the protein target, they may not always translate to the corresponding therapeutic or so-called phenotypic activities above the molecular level. And that is because the biological system is a very complicated system. Beyond the molecular binding, then there are so many other factors in the cell, in the body, that affect the final result, which is beyond the target-based virtual screening can predict. And we often, see this type of discrepancies in literature, and sometimes also say that in our own project. So, for example, sometimes even there's a hit that successfully co-crystallize with the target protein. So there's no doubt it can really bind to the protein. But then if you test that hit in the phenotypical assay, let's say a cell-based anti-tumor assay, the hit is not active in terms of killing the tumor cells. Then that comes to the second approach, which is phenotypic-based virtual screening. So opposite to the target-based, phenotypic-based, it refers to the phenotypic activity, also called the therapeutic activity. So then using this method, of course, you are more likely to achieve the therapeutic effect you want. However, if you only use a phenotypic-based virtual screening only approach, then there's also this downside that often those phenotypically active hits, they don't have a clear molecular mechanism, and that can increase the risk of attrition in later stage of development or even clinical development. For example, there may be some hit that looks like phenotypically active, but it's not really targeting a protein target that is clinically relevant, then that may result in the failure of the project in the future. And because of this, the best way, of course, is you combine both the phenotypic-based virtual screening and the target-based virtual screening together for your research. Then it's more likely we will have the hits that are both target-specific and phenotypically potent. And then there are those specific tools or methods that we use for each type of the virtual screenings. And I briefly divided those tools or approaches into two categories. One is called a simple system method, one is called a complex system method. So let's talk about the target-based virtual screening first. And here are some tools that we often use for our program. And first tool, of course, is the ligand target docking using classical or quantum mechanisms. So that is a very popular way to predict protein binding specificity. And this is a simple system method. It is because in this method, we use those physical equations to describe the interaction of the ligand and the target. So in physics and mathematics, people always try to use a simple equation to describe everything. And the simpler the equation, the more beautiful it is. However, there's at this current technology level, there's some limitation about applying the simple system method. It is because at least for two reasons. One reason is that for some systems, we haven't found the simple equation that can describe every behavior in that system yet. And the second reason is that even for some system, we had the perfect equation, but to get a very accurate, realistic calculation result, there are so many parameters, requires so much computing power that the current computer technology cannot handle. So therefore, sometimes we have to come to the so-called complex system method. In comparison with the simple system method, the complex system method, so it's not about finding the simple equation. Rather, we just leverage the artificial intelligence to observe the system and then let the AI learn based on those observations and then make new predictions. And then two popular approaches for target-based virtual screening or prediction. One is AI-driven docking. It's similar like the conventional docking using classical or quantum mechanics, but instead it's a train. It's AI-driven docking refers to we train the AI models based on non-protein ligand interaction data and protein ligand complex structures. So there are some famous examples, like one recent one is this so-called Rosetta fault. So that is an algorithm developed by Dr. David Baker, and he actually recently shared the Nobel Prize with the developers of the AlphaFold. And so besides this AI-driven docking, that is on the atomic level, there's also this molecular level prediction that the AI model can do in that way. So instead of you train the AI model with the ligand protein structure, you train it with protein binding assay data. So it can predict whether a new test article can bind to the same protein or not. So that's some popular method for target-based virtual screening. Then when it comes to the phenotypical-based virtual screening, because that is at least a cell system or even more complicated system, so there's no single equation we found to describe them. So it's all complex system method. And there are at least three popular methods we often use in our project. One is the AI models trained with cell-based assay data, so that can directly use to predict cell-based phenotypical activity. And one popular algorithm we use for that is explainable deep neural network, which I'll discuss a little more technical details in next slide. So the second popular method is instead of you train the AI model with cell-based assay data, it can also be trained with ex vivo or in vivo assay data. And thirdly, there's also those large language models trained with medical literature and other type of data, which can also do those type of predictions. So the large language model is a new concept. So far, it's not the result is not highly useful yet, but I believe it's a future trend. In the future, when we develop more useful large language model, it will play a more and more critical role to help our drug discovery. So yeah, that's the overview of those methods. And here, I think this is just some more technical details of one of the methods we use. That's explainable deep neural network models. So we often use this type of algorithm to build models for the structure-activity relationship prediction. And in that practice, there are 4 steps involved. The first step is prepare the training data set. And because this is a structure-activity relationship model, so of course the training data will contain 2 parts, the structures and their activities. Speaking of the structures, if it's small molecules, we usually will need to find a way to turn those structures into a string of numbers so that it can be processed mathematically. And one popular way to do that is called molecular fingerprints. And I listed a Journal of Chemical Informatics Modeling paper published in 2010 that has more detailed description of the molecular fingerprints. But anyway, use that you can interpret those chemical structures numerically. So as Paige mentioned earlier, besides small molecule, actually there can be other drug modalities in HTS, for example, RNA drugs, antibody or peptide. So if it's those type of test articles, then instead of chemical structure, it will be the sequence of those test articles. And similarly, there's those natural language processing algorithm can turn these sequences into a string of numbers, so it can be processed later. So besides the structure, the next thing, of course, is activity, and here is more about the phenotypical activity. And the most popular value we use for the phenotypical activity is the IC50 values. And thanks to the high throughput of the screening technology, then we can have IC50 measure for a large number of compounds that good enough to train the AI models. However, besides those IC50 numerical values, there can also be category types of activity. So let's say sometimes in high-throughput screening, there's just a flat curve. So then, of course, you wouldn't get the IC50 value, but instead, maybe the value will be activity greater than 20 micromolar, something like that. So those will be treated as a category type of activity data. But besides that, there can also be other types of category data. So let's say if we study genetic disease, then some compounds, they are active against one mutation, but not the other mutation, then those can also be used as to denote the phenotypical activity. Or if it's an infectious disease research project, then maybe some test article active against one viral strain, but not the other. So those can also be used to label the activity of the test articles. But anyway, once we prepared the training data set, the second step is you train, let the deep neural network to learn the relationships between those structures and activities. So, and again, I also listed chem reference. There's more mathematical descriptions of those algorithms. So just to keep it simple, once you let the model learn the relationship, next step, the third step, then you can use this model to predict new molecules of interest. And similarly, first you turn their structures into a number of strings so that it can be input into the model for the prediction. And then the last step, the fourth step, you will get the predicted results. So here you can see that we have a new molecule and the predicted IC50 is 2.5 micromolar. But because besides this whole molecule property predictions, another good thing is that we can also let the model to predict the per atom contribution. Once we have the per atom contribution, then we can let the model explain the whole molecule prediction result. So as showing on this colored finger of the molecule, so the model can color different part of the molecule. Some part in green means the model think is active, and some red part is the model think is inactive. Then there's two good things about this explainable results. First, if in next step our chemistry team want to develop new analogs, then based on this colored molecule, they know that they need to keep the green part, but then modify the red part to improve the potency. And the second good thing is that this can also help us to better judge the accuracy of the or reliability of the prediction result. I mean, of course, the model will give you a confidence score, but that is a theoretical score, may not always correlate with the real world accuracy. But then based on these explainable results, we can leverage our medicinal chemistry knowledge to say that whether we think the explained result makes sense or not. If it obviously not consistent with some knowledge we already know, then maybe we need to doubt whether the result is reliable or not. So after all, to judge the real-world accuracy of those AI models, we still need the expertise from human scientists. So yeah, that's some technical details. And leveraging those approaches I described before. So we are doing this AI-powered HTS. So this is a very brief workflow for that. We will combine the phenotypic virtual screening and the target-based virtual screening together to prioritize the HTS library, and then those prioritized library will go through all the HTS process, and eventually we will get some hit candidate for further development. And those candidates, they are very likely to be both target specific and phenotypically potent, so very promising scaffold for the future development. And Paige will talk about more details about this workflow.
Yeah, and I just wanted to briefly give more detail about the front end of this and sort of what it looks like. So as Sixue explained, different tools and different methods are applied in silico, as shown with the AI icon in the upper left, and that informs what we are going to screen. So that powers our HTS. So if our libraries are bound to plates, we would prioritize plates that are enriched in those active compounds that have been identified, or if we have tubes, compounds individually stored in tubes, we can use automation to pick those and create basically custom sets to confirm. And this can occur depending on the number of the compounds and the format, either in a single concentration or in a concentration response. Of course, it's nice to have sort of this ability to do an enriched set and screen fewer compounds, because then we can we're more able to do the more intense concentration response to give the IC50 or EC50 potency values. But we can still use single point data and to categorize things. And then this feeds back into the model, and it can be an iterative process that both powers the HTS, but then also improves the models that are used. And Sixue is going to go over some examples of how we've applied this here at Southern Research.
Yeah, thanks. So yeah, this briefly shows some examples that using this AI-powered HTS, we can really increase the hit rate. So there's two specific examples. They are the alpha viruses and flaviviruses projects we have. Those viruses, they are both tropical viruses that currently lack effective treatment. And so for the alpha viral project, previously we had a large-scale HTS campaign. That is before we applied the AI method. So we screened 150,000 compounds, and we have the hit rate of 0.16%. So, however, this overall is a very successful campaign because we have 4 leads in development out of that screen, and one of them is even in preclinical stage. Based on that previous success, and also leveraging those abundant HTS data, we recently did another AI-powered HTS to try to find additional lead molecules and hit scaffolds to further develop. And then we screened 50 AI prioritized compounds, and the new hit rate is 8%. You see it's obviously higher than using unprioritized library. And what's more important is that those new hits, they are both phenotypically active and has a clear target. That's for the alpha viral project, the target is the viral NSP3 protein. And similarly, in the second example is a flaviviral project. So this viral actually is more sensitive to the drug treatment than the alpha viral. So you see that overall the hit rate are higher. So for the previous HTS is 0.3%, and the new AI-powered HTS is 54% hit rate. And again, the hits, they are both phenotypically potent and has a clear molecular target. So especially the flaviviral target has already been experimentally validated by our collaborators in Oregon Health Science University. So besides those two examples that we only conducted a small scale AI-powered HTS, we actually also do this AI-powered HTS in larger scales. That's, for example, in a new project we just started. The scale of the AI prioritized library is 10,000 compounds. So currently the project is ongoing, and let's see if there's anything exciting out of the project. So lastly, and we had some considerations about this AI-powered HTS concept. So from the in silico perspective, again, it's better we combine both target-based virtual screening and phenotypic-based virtual screening together, so that we can increase the chances that we find some hits that both are target specific and phenotypically accurate, phenotypically potent. That provide more promising scaffolds for future development.
Yeah, and I wanted to point out that in silico methods can produce thousands of hits to confirm experimentally. So, there's still a need for being able to run, automated plate-based assays. And just some practical considerations, if you have access to an in-house library that's bound to plates, as we've described, choosing plates enriched in hits is an efficient, economical way of confirming hits and further informing the model. If you're building up a compound collection, consider a dual format, maybe tubes and plates, or if you're ordering, you may order the compounds that are hits, but that can provide flexibility to create custom sets defined by in silico efforts. So you can have these sort of customized sets that are the results of the in silico campaigns. And also just always consider the impact on your screening method, on your analysis method, especially if you're typically beginning with the null hypothesis that compounds are inactive. So you need to consider how you're doing that, especially with single concentrations. And also want to point out that if there's an absence of one or both of these capabilities within your organization, it doesn't necessarily mean that you have to exclude their use. Drug discovery is team science and reaches across organizational boundaries, as we've shown with a lot of the different collaborations that we have with different organizations that bring their expertise to the table. So with that said, we want to acknowledge a lot of people that are part of that team at Southern Research. Too many to name, but several of them are listed here across different departments, as well as our collaborators, again, collaborations across the country, and our funding sources, National Institutes of Health. We have a really a great program with the Cystic Fibrosis Foundation in collaboration with UAB. Our Center for Clinical and Translational Science, which is an 11 institute and university consortium funded by NCATS, that the hub of that is at UAB, and our Alabama state-funded accelerator program. And there's contact information here at the bottom. And lastly, I just want to say, just as part of the wider drug discovery community, as Heather mentioned earlier, I'm honored to serve as president starting now and going into 2025 for the Academic Drug Discovery Consortium, which is a nonprofit organization dedicated to building this community of universities and nonprofits and government agencies and companies that support and partner to make drug discovery happen. So you can follow us to learn more about upcoming events. So with that, we'd like to thank you for your attention, and we look forward to answering your questions.
Thank you so much, Dr. Vinson and Dr. Zhang, for your wonderful and informative presentation. It looks like we do have a few questions here. We'll start off with Matthew. He asked, can we replicate this prediction at our end with the same source code and data?
Yeah, so looks like it's more of a in silico question. So yes, for sure. Some of the projects we are preparing publications, and if we have a publication involve the use of AI model, we usually will also publish the codes, and they will be deposited to GitHub that people can freely use, as well as we will also upload the training data set to GitHub. So if you use the same training data set and run the same code, you will be able to replicate the results. So there are some exceptions that we have a few projects because we haven't applied a patent yet. Some of the chemical structures we included in the training data set are confidential. So for those, we don't have publication, and we cannot disclose them currently, but in the future, once we have patent, they can be disclosed, so they will also eventually become available. But if the training data are from literature, there's no problem. Thanks.
Okay, thank you for that. We also have a question. It's like a three-part question from Weiji. What are the limitations of AI HTS? That's his first question.
Limitations. I mean, I think there could be several, right? I mean, a lot of times you go, and it's probably not unlike other drug discovery campaigns where you're using whatever method to identify hits, or even to inform the follow-up chemistry. So you're limited in, are your assays representative, or are they going to translate into the clinic? So if you're talking about limitations in terms of what could be barriers to success, whether or not your target is truly valid and that will translate into the clinic, that's one. I mean, it wouldn't matter what method you use. You're going to have a limitation with that. If you're talking about scale, I would say the limitation would then be the experimental methods to test the in silico, because although, and correct me if I'm wrong, Sixue, but there are certain in silico methods that are more intensive and would take longer to generate the data, like probably the complex versus the simple, but you're still accessing this large chemical space. So then it's really probably the throughput of the assay and the techniques you're using to confirm those and test them. And also maybe the availability of the training set. But what we like, one of the reasons I brought up the history of our center is that we now have some bioactivity data that can inform a lot of the models that Sixue has developed. So that could be a limitation in terms of being able to have that training set. Do you want to add anything to that?
Yeah, I think Paige had a very well summary, and I would like to further comment on the training data set problem Paige mentioned. So far those AI model, their accuracy is based on the quality and the abundance of the training data. The more abundant the training data, the better the accuracy is, as well as the quality means that in some data, like if it was repeated in different labs, the results are different, then those there's already larger experimental error in those data. If you use this data to train the model, your model also have those errors. So the quality and abundance of the training data determines the determines the accuracy of the model, and that's one of the limitations. So hopefully in the future, there can be those next generation AI that are less relied on those the training data.
Okay, awesome. Thank you. The other part to the question, so we have quite a few questions here. So I'm going to go to the next person, and it says, what is behind the AI-powered HTS? Is it similar to ligand-based screening? Similarity? What is the size of the training data set? And AI enhanced the hit rates, but what is the diversity of new hits?
Yeah, that's a good question. So in terms of phenotypic-based virtual screening, it is ligand-based, and I mean, if it's also like if it's biological drugs, so that will be based on the sequence of those biological drugs.
Okay. So we have another question. Do you find docking or binding assay data models to be more effective? For binding assay data, do you ever use primary screening data such as percent inhibition instead of IC50s? And how do you deal with the prevalence of false positives and false negatives from primary screen data that also exist in DELs?
Yeah, that's also a good question. So the first part of the question, I guess you would like to ask whether docking and protein binding, which one is more reliable? Is that the question?
The first part.
Yeah.
Do you find docking or binding assay data models to be more effective?
Oh, so that actually I think is both can be good approaches. So of course we usually use docking because not every project you have a lot of protein binding assay data that you can train the AI model. But if you have that data, it's good you use both docking and those AI model trained with binding assay data. And then another question is that is about like what type of binding assay data we use. So this can be either in-house data or from literature, and we would prefer numerical value, which means the IC50 value, and in some binding assay data, it's the KD, so dissociation constant. So that is also fine. They are also numerical values. But sometimes it may also be just category value, say it says active or inactive. So that will be a binary category model with two different groups of compounds. That is also fine if you don't have other types of data. And then the last part of the question is about the false positive. So we have this practice we do for every project. It's called pan-assay interference compound filtration. So there are literature published about the pan-assay interference structures. So those are structures that are widely acknowledged in the field that they cause false positives in many assays. For example, they may be self-fluorescence compounds or very reactive compounds or metal chelators. So we have cheminformatic filters for that. We will remove those compounds. They wouldn't be in we don't use them in our training data, and we of course wouldn't suggest hits that are those type of compound to our chemistry teams. So besides those pan-assay interference, different assays, they may have specific interference issues. So let's say if it's luciferase assays, if compound inhibit luciferase protein itself, it may be a trouble. For that, we have some prediction method. Let's say there's a luciferase crystal structure. You can dock the compound to see if it likely bind or not. Although the prediction wouldn't be always accurate, but it's a a part of the effort to help reduce false positive. Thanks.
Okay, thank you. We have we have time for a few more. Are there publications on this prediction? If so, can you send the link of the publication?
Yes. So but then how should what's the way to share those?
Could you
If
add
you
those
want,
to the biography that's published on the webinar site, perhaps?
Yeah.
Jasper, can we do that?
Yes.
Okay.
I mean, is that
Right. So
acceptable?
yes. And do you also, I think, do you mean our own model or some reference from literature? Because for our own model, if it's still confidential, then we cannot directly share. But you are welcome to reach to us to talk about the collaborations. Once those models are officially published in any manuscript, then of course in those manuscripts there will be the link to the model.
Okay. Another question, why choose DNN algorithm? Is it more accurate than CNN or GNN or other DL algorithms?
Yeah, that's also a good question. So actually, so far there's no evidence to prove that one algorithm must be more accurate than the other one. For example, besides the deep neural network model algorithm, we sometimes also use a graphic neural network algorithm or other algorithms. So one practice, if we sometimes do if we don't feel the model from the deep neural network is very accurate, then is that we will build a model using different algorithm and compare the accuracy, and then eventually we will choose the most accurate algorithm for the final model. And in some cases, if each algorithm has their own unique outcome. For example, one model you have more false positive, the other one you have more false negative, something like that. Then we also do an ensemble model, which we combine the prediction result from multiple algorithms together to have a consensus model.
Okay. Are the phenotypic AI models used mainly for PK/PD properties, or do you also use cell-based screening datasets?
So the phenotypical activity, the most often cases are the cell-based assays because those are cheaper, faster than the in vivo assays. So usually there are more data from cell-based assay that you can use to build the model. But there are some cases that there's indeed enough in vivo data to build the model. One case is a model we use to predict the clearance of the drug in mice. So for that, there's enough data from literature that we can use.
Okay. Looks like we can do a few more. Is it possible to further select the hits by using the available data and models to narrow the load of HTS test plates? Excuse me.
Can you repeat that, Heather?
Sure. Is it possible to further select the hits by using the available data and models to narrow the load of the HTS test plates?
Narrow the load?
Narrow the load, yeah.
Okay. I mean, that is partly, I think, what we're doing. You know, we can you know, do some initial screening, and instead of doing maybe, you know, all 750,000 in our collection, we can have what we select prioritized. And, you know, if we're doing some confirmation or even just initially doing things in concentration response, we don't really do sort of ultra HTS here, but we are able to run a concentration response on a pretty large number of plates, and even maybe counter screen in parallel. So if we're doing that process and it's iterative, we can, select some, initial hits to purchase and, reconfirm sort of in a real time, sort of semi-real time. And what we really want to do is, shorten the time to, getting to a lead candidate. So I think if I understand the question correctly, it's yes, and that's really part of what we're doing in this process.
All right. Well, it looks like we are out of time for questions. If you did have a question that we did not get to, these questions will be sent over to Paige and Sixue so that they can see these questions and they can respond back to you. But thank you very much for all of the questions, and thank you to both of you for your insightful answers. We now have a surprise giveaway announcement. So we have a year-end celebration going on with 20% off all of our products, excluding our inhibitors. And then we have an exciting gift unveiling soon, which is our keychains, which you can see here. So they represent Black Friday, Christmas, and Green Miracle. And as thank you for your participation, we have prepared a surprise giveaway for all attendees who register as MCE website users. So please register as soon as possible. Thank you for attending today's seminar. Thank you to Dr. Zhang and Dr. Vinson. And we look forward to seeing you at our future events.